Hello dear students. Today I am going to discuss about the topic of enzymes and its production, which is included in the first unit of your syllabus. Hopefully this is the last topic from your first unit. What are enzymes? The enzymes are macromolecular biological catalysts. What is the main function of the enzyme? They are mainly used to accelerate or catalyze the chemical reactions. The molecules at the beginning of the process are called as substrates, which is denoted as capital S, and as the enzyme converts them, they become the products, which is denoted as capital P, and enzymes are denoted as capital E. So, you must have gone through that reaction. E plus S gives you ES, then E plus P, and the product is separated. The microbial enzymes are the biological catalysts for the biochemical reactions leading to the microbial growth and respiration, as well as to the formation of fermentation products. In this particular topic, we are going to mainly lay emphasis on the microbial enzymes. Okay, so here you can see I'm trying to give you a 3D structure of the enzymes. There are three colors which have been used. One is gray color. You can see that the gray color is showing the scaffold to protein structure, showing which is the scaffold to support and the position of active site. Okay. Now, which are the active sites? There are two types of active sites. One is the binding site, another one is the catalytic site. The binding sites are shown in blue color, and you can also see this black color substrate has already been attached to it, and the catalytic site is shown in the red coloration. There are two types of enzymes. One is adaptive, another one is constitutive. The adaptives are produced only when the need arises. For example, when a cell is deficient of a particular nutrient, then an adaptive enzyme is produced. Constitutive is always produced and they are not dependent upon the amount of substrate, unlike the adaptive. Now we are going to study a little bit of historical background of this enzyme production. You can see here I have mentioned that the first enzyme produced industrially was the fungal amylase tacadiastase, which was employed as a pharmaceutical agent for digestive disorders. This is an important question for MCQ. Okay. Due to the occurrence of allergies among the production and co-workers and consumers, the sale of such enzyme utilizing detergents was decreased drastically. Now, which enzyme was used for this particular detergent activity? That is protease. Mainly protease was used. So, in order to overcome the problem of allergies and all, the special technique of microencapsulation was adapted. I think you have studied about this microencapsulation also in the immobilization of enzyme topic. By adapting this method, the production of the enzyme was made risk-free for the consumers as well as for the workers. The microbial rennin is also produced, and which can replace the calf's rennins, which is used for the cheese production. Now, what are the different location of enzymes? There are two types of enzymes occurring. One is endocellular enzymes, another one is exocellular enzyme. Endocellular enzymes which are produced within the cell. From the name itself, it signifies endo, is within the cell or the cytoplasmic membrane. And exocellular is which are liberated in the fermentation medium, which can attack large polymeric substances, and examples are amylases and proteases. What are the different improved prospects of enzyme application? The first one is microbial genetics. It resulted in high yields by the process of genetic manipulation. For example, Hansenula polymorpha, it is one example, pay attention, has been genetically modified so that 35% of its total protein consists of the enzyme alcohol oxidase. Now, these both are related. You need to pay attention for MCQ type question. The next is optimization of fermentation conditions, such as the use of low cost nutrients, optimal utilization of components in nutrient solution, the temperature and pH. Other points are that the new cell breaking methods like homogenizer and bead meal and sonication have been adopted. The modern purification process of counter current distribution, ion exchange chromatography, means different type of chromatography, and also the process of precipitation by using the solvents such as alcohol and acetones have been adopted. I have also shared a handwritten notes with you people. You can see there that I have given in these details in the method of enzyme production, particularly in the recovery process. Okay. Next is immobilization of enzyme, which you all are well aware of since we have already completed this topic. The next one is continuous enzyme production in special reactors, which is also called as bioreactors or fermenters. Now, what are the different? Now, we are going to emphasize on the particular method of enzyme production. There are two methods. One is semi-solid culture, another one is submerged culture. Okay. So, in the details of the semi-solid culture, I have given it in a flowchart form. You can see that the enzyme producing culture is grown on the surface of a suitable semi-solid substrate. It can be a moistened wheat or rice bran with nutrients, means I am talking about the media. Next is it goes for the preparation of production medium, which might contain bran, which is mixed with solution containing nutrient salts such as NaCl, KOH, etc. Then we go for the maintaining of pH at neutral level. Medium is steam sterilized in an autoclave while stirring. You all know how to sterilize by using an autoclave. The next step is the sterilized medium is spread onto the metal trays up to a height of 1 to 10 centimeters. Okay. The culture is inoculated either in the autoclave after cooling or in the trays. Like this, the culture is inoculated using a inoculation loop. Then the high enzyme concentration in a crude fermented material is and you obtain the produced enzyme. The enzyme is produced. Okay. Here I have given a tabular data of the different types of enzymes or the examples which can be produced by semi-solid culture, such as alpha amylase, the microorganism or microbial strain used is Aspergillus oryzae, glucoamylase, Rhizopus species, lactase, Aspergillus oryzae, pectinase, Aspergillus niger, protease, Aspergillus niger and Aspergillus oryzae, rennet for mucor bacillus. Okay. What are the different advantages of this particular semi-solid culture method? That it involves comparatively low investment, low cost, allows the use of substrate with high dry matter content, hence it yields with a high enzyme concentration in the crude fermented material. To cultivate those molds which cannot grow in the fermenter due to wall growth, that can also be grown in this type of semi-solid culture. It also allows the mold to develop into a natural state. What are the disadvantages? It requires more space and more labor. Because it is open, the trays are open, it involves a greater risk of infection. It is also difficult to introduce automation in such system, means it has to be done manually every time because it needs skilled persons. So, automation of this type of process, semi-solid culture process, is difficult to introduce. Next culture technique is submerged culture. Okay. In this, the fermentation equipment used is the same as in the manufacture of antibiotics. It is a cylindrical tank of stainless steel and it is equipped with an agitator, okay, an aerating device. This is actually basically a fermenter, okay, a cooling system and various ancillary, that is the foam control, pH monitoring device, temperature, oxygen tension, all these are controlled by the different types of ancillary equipments. The growth is not enough to obtain a high enzyme yield. The presence of inhibitors or inducers should also be checked in the medium. For example, presence of lactose induces the production of beta-galactosidase. As the inducers are expensive, constitutive mutants are used which do not require an inducer. Okay. The glucose repress the formation of some of the enzymes, thus the glucose concentration is always kept low. Either the glucose can be supplied in the incremental manner or a slow metabolizal sugar can be supplied for the production via this particular method. There are certain surfactants also which can be used as a production medium which can increase the yield of certain enzymes, and certain non-ionic detergents such as Twin 80 and Triton which are also frequently used. What are the advantages of this particular method? It requires less labor and space, there is a low risk of infection, and automatically controlling this type of process is very easy. The disadvantage of this particular method is that initial investment cost is high, means the setup and all the fermenter purchasing, the setup of the particular fermenter. It is initially the cost is very high, but once it is set up, it is easier to use since it is an automated process. Next is, what are the steps after the fermentation? Once the fermentation is finished, the fermentation liquor is subjected to rapid cooling to about 5 degrees centigrade in order to reduce the deterioration. Then the separation of microorganism is accomplished either by filtration or by centrifugation of the refrigerated broth by adjusting the pH. And to have a high purity of the enzyme, it is precipitated with acetone and alcohols or inorganic salts such as ammonium or sodium sulfate. If you are going for a large scale operation, salts are preferred to solvent because of the explosion hazards. So that's for this particular topic. I have already shared with you a research article, one handmade note, and then I am giving you this lecture video. You can go through this again and again, and if there is any doubts, you can ask me via the Google Classroom. Students, we really need to work hard because we are in a condition where we are not able to take the class physically. You people are also at home due to this coronavirus. So I hope I will get proper cooperation from your end. I am ready to help you at any cost. You can clarify all your doubts without hesitating. Thank you.